Hello friends, today in this video we are going to discuss about the first 5 steps of glycolysis. As you know, there are a total of 10 steps in the overall process of glycolysis. We can divide it into 2 phases. First 5 steps is first phase and then second 5 steps are in second phase. Okay. In these 5 steps, it prepares to release energy. So, in these 5 steps, this is energy investment phase. Energy is being invested so that after these 5 steps, we can get energy, we can generate energy. So, this first 5 steps is also called as preparatory phase or energy investment phase. Okay, and the next 5 steps are the payoff phase. Okay, these 5 steps will pay off the energy. It will generate energy. So, this second 5 steps are also called as payoff phase or energy generation phase. Okay, let's discuss about the first 5 steps, that is the preparatory phase. Okay, so as you know, in the process of glycolysis, first the substrate is glucose and ATP. Okay. In the first step, what happened? In the first step, glucose is converted to glucose 6-phosphate. Okay. By the help of the enzyme hexokinase, you have to remember this. Okay. Hexokinase and ATP is used here. ATP is converted to ADP and PI. Okay, here a metal ion, that is Mg2 plus, is also used. You have to remember all these things: the hexokinase enzyme, the ATP. You should also remember the structure of glucose. Okay, the ring structure of glucose. And you have to note one thing, that this is an irreversible step. We cannot go back from glucose 6-phosphate to glucose in this step. In this preparatory phase of glycolysis, we cannot go in reverse direction from glucose 6-phosphate to glucose. First step is finished, then the second one. In the second step, what happened? Glucose 6-phosphate is converted to fructose 6-phosphate. Okay, and this is a reversible reaction. You can again go back from fructose 6-phosphate to glucose 6-phosphate. And the enzyme used is phosphoglucoisomerase. Here it is a isomerization reaction. Okay. So the third step is again a kinase enzyme is involved here. You can note that phosphofructokinase. In this step, fructose 6-phosphate F6P, it is the small letter. Okay, fructose 6-phosphate is converted to fructose 1,6-bisphosphate FBP, fructose 1,6-bisphosphate. And you can note that this is also a irreversible step. This is also a irreversible step. Okay, here the enzyme used is phosphofructokinase. You have to remember it. Okay, revise it again and again so that you should not forget it. Okay. After that, the fourth step is regulated by the enzyme aldolase, where the fructose 1,6-bisphosphate is converted to glyceraldehyde 3-phosphate, that is G3P. Here you can see the structure, molecular structure of glyceraldehyde 3-phosphate. In the 3 carbon position, there is a phosphate group here. Okay. And after that, the fifth step is regulated by the enzyme triose phosphate isomerase. Again, a isomerization reaction is here. Okay, where the glyceraldehyde 3-phosphate G3P is converted to dihydroxyacetone phosphate. Okay, here you can see the molecular structure. We have discussed the 5 steps of glycolysis. Okay, but after that, what happened? This dihydroxyacetone phosphate again is converted to glyceraldehyde 3-phosphate, as it is a reversible reaction, as you can see here. Okay, it converted to glyceraldehyde 3-phosphate, and then in the sixth step what happened, this glyceraldehyde 3-phosphate undergo this change. Okay, thank you friends for watching this video. In the next video we will be discussing about the next 5 steps of glycolysis. Thank you.